Hello everyone, my name is Steve Blackwell. I am the Conservation Collections Manager at Desert Botanical Garden, and I am here to talk to you about a project that we're working on with this little plant. This is the Canelo Hills Ladies Tresses Orchid. It is native to southern Arizona, and it is an endangered species known to only 2 remaining populations. And one of the issues with it is when we're trying to find these plants out in the wild is it's very difficult for us to locate. As you can see, it's very well hidden amongst all of these grasses. And so what we've done is we've enlisted the help of specially trained scent detection dogs to help us locate these plants. The reason why we're interested in this plant is because we want to be able to collect seeds from them in order to propagate them back at the garden. Now, in order to propagate them, typically what you need is a very specialized fungus which allows these seeds to germinate. But if you don't have that fungus, what we could do is we can actually provide the media and the nutrients that these seeds need to germinate in the laboratory. And that's what we've been doing so far. So we're back out here again trying to see if we can find more of these plants so that we can collect additional seeds to take back to laboratory and propagate some more plants.
I'm Laura Lee Oliver. I'm a dog trainer. I own Canine Incentive LLC. We're a dog training company, but we specialize in training conservation detection dogs. I'm out on this trip looking for a rare orchid species. The 2 dogs that are working on this orchid project are Circe, she's a Labrador Retriever, former agility dog turned conservation canine, and the other dog is Muon, and she is a Belgian Tervuren Malinois cross who I got from rescue in the Utah area. So most of the dogs that we work with, we get from local rescues or shelters, or sometimes they're from a rehome, from a family that maybe couldn't handle their high energy, because they certainly have it. So those are the 2 dogs on this project this week.
Once we're able to determine whether these dogs are able to locate these plants, what we're going to do is take them to previously known populations where they haven't been seen for several decades, and we're going to see if they can try to relocate them there. So the genus of this plant is Spiranthes, and you can actually see why it's called that. You can see how the flowers spiral around the inflorescence.
I got interested in this orchid because I've been working on wetlands for over 15 years, and wetlands are one of the rarest habitat in Arizona. This orchid is only known from 5 localities in Arizona. Only 2 locations right now that we've been monitoring their populations the last few years. We're trying to understand why in some years we can find over 200 of these plants at a certain site, and the next year follow up and we only see about 40 or fewer plants at a site. But this is very common in a lot of orchid species. For the populations to fluctuate wildly, in some years they might not even emerge. We're working with the North American Orchid Conservation Center to preserve the orchids in Arizona, and one of the things that we're doing is collecting seeds from the wild orchids and trying to isolate the fungi from their roots so that we can grow them in the laboratory and then maybe reintroduce them into the wild populations. But also just to understand their biological functions with each other. So with the North American Orchid Conservation Center, several of us at the garden are going around Arizona and collecting orchid seeds and the roots from the orchids so that we could send those roots to the Smithsonian where they can isolate the fungi that inhabit the roots.
I'm Dennis Wiggum. I'm a senior botanist at the Smithsonian Institution. I work at the Smithsonian Environmental Research Center, located near Annapolis, Maryland, and I study a wide range of plant-related ecological issues. But in the context of why we're here in Arizona, it's related to the North American Orchid Conservation Center, which I started about 10 years ago with the goal of assuring the survival of all the native orchids in the United States and Canada. There had not been a national effort of that type. The connection to the Desert Botanical Garden is through Andrew Salawan. Andrew was one of the first folks who came on board with our collaborative effort to assure the survival of all the native orchids. And the thing that brings the native orchid issue to bear for this particular project was the fact that I'm on a board of directors of an organization called Biophilia, which is based in New Mexico, but they work across the country. And here in the desert Southwest, they're involved in efforts to find ways to manage the landscape, to keep water on the landscape for longer periods of time. That's clearly a big issue out here. what caught my attention was the fact that doing that would create habitats that would be beneficial to both the orchids and the fungi that the orchids need. So that's what brought us together, and what we're doing on this trip is locating sites that have native orchids, and we're collecting samples from those orchids, but we're also putting in equipment to monitor the soil moisture and the temperature of the soil, two factors that are important not only for the orchids but also for the fungi that the orchids need.
My name is Melissa McCormick, and I'm an ecologist with the Smithsonian Environmental Research Center. And I do a lot of research on understanding the interactions between orchids and their mycorrhizal fungi and orchid ecology in general. So all orchids, as just a part of their natural life history, require associations with fungi that are often very specific fungi that they need to digest in order to get their food. So most plants associate with mycorrhizal fungi. These are fungi that grow into the roots of the plants, and they help the plants to get the nutrients they need and soil moisture. But orchids sort of take that another step. They actually digest their fungi, and so as a result of that, they absolutely depend on these associations with fungi in order to grow from the very tiny seeds that they start out with into a full-size plant that then can reproduce. And so what we're doing as part of this project is we are collecting samples of orchid roots to identify the fungi that different individual orchids are using to determine whether the same fungus is used from one population to the next. And then we're going to be taking soil samples to understand where that fungus is in the soil. And the reason we have to use DNA to do that is because if you take an amount of soil about the size of a lima bean, you probably have 150 species of fungus in that soil sample. And so to figure out where the soil, the particular fungus your orchid needs is, you have to use something like a specific probe that just matches to that fungus. So what we'll do is with these leaf samples and these root samples that we're collecting this trip is we will take them back to the lab at the Smithsonian, where we will extract the DNA from them and use that to, as I said, we'll also try to isolate the fungi from the orchid roots, and those fungi will then be used with the orchid seeds in order to support germination that would allow us to potentially grow these orchids in the lab and be able to reintroduce them to the natural environment to help supplement the natural populations. We'll then also use the DNA from the leaves to identify the genetic diversity, from the soil samples to identify where the fungi are present in the natural environment.
The challenge that we had is that the species that we were given to train the dogs on was not the same one that we're actually looking for in the field. Since those are protected, we couldn't actually have them at my training facility, so we trained on a different species. So our challenge this trip was to see that the dogs would associate the target orchid with the one that we trained on, which I'm happy to say they did.
Right in here.
Right in there. Yeah, right in there.
Watch your little cute feet. Like in this area here. Just shoot.
Sorry, baby.
Here, sweetie. Keep working.
How the hell are we supposed to find orchids in this mess? Right? Go ahead, slosh down. Go slosh around. Gotcha.